Hello everyone, here we are to explain about transcription process in detail.
Transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. It is governed by the principle of complementarity with one exception that adenine now forms base pair with uracil instead of thymine. In transcription, only one DNA is copied into RNA. Both the strands are not copied during transcription. If it acts as a template, they would code for RNA molecule with different sequences. And if two RNA molecules produced simultaneously would be complementary to each other and will form double-stranded RNA. Transcription unit. It consists of three main regions, a promoter, the structural gene, and a terminator. Promoter is the starting point of a transcription process and it is present upstream frame end of the structural gene. Structural gene is a part of the DNA strand that has a polarity of 3' to 5'. Terminator is located at the 3' end downstream of the structural gene.
Types of RNA. In a bacteria, there are three different types of RNA, namely mRNA, tRNA, and rRNA. All these three types of RNA is required to synthesize a protein cell and they are having functions of their own. mRNA. It is the messenger RNA and provides the template. tRNA. It is the transfer RNA and brings amino acid and reads the genetic code. And rRNA. It is the ribosomal RNA and plays structural and catalytic role during translation. Process of transcription in bacteria or prokaryotes.
The process of transcription in a bacteria are initiation, chain elongation, and termination. Initiation. The RNA polymerase along with sigma factor or initiation factor binds with the promoter region of DNA and initiates transcription. It helps in the opening of DNA helix and separates a strand of DNA. Chain elongation. RNA polymerase builds a single-stranded RNA using one of the strands of DNA as template, using nucleoside triphosphate as substrate and polymerases in a template-dependent fashion following the rule of complementarity. It somehow also facilitates the opening of helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme after this. It takes place in 5' to 3' direction and after this, sigma factor dissociates from RNA polymerase and can be reused. Termination. Once the RNA polymerase reaches the terminator region, the newly created mRNA along with RNA polymerase and also termination factor or Rho factor falls off. This results in termination of transcription.
Process of transcription in eukaryotes. Transcription in prokaryotes occurs in cytoplasm, whereas transcription in eukaryotes occurs in the nucleus. In case of prokaryotes, transcription is catalyzed by a single RNA polymerase, whereas in case of eukaryotes, transcription is catalyzed by at least 3 RNA polymerases, RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. There is a clear-cut division of labor between RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 transcribes ribosomal RNAs 28S, 18S, and 5.8S. RNA polymerase 2 transcribes the precursor of mRNA, that is heterogeneous nuclear RNA, which is processed into an active mRNA from its inactive form by splicing, capping, and tailing, which is a difference between transcription in eukaryotes and prokaryotes because in case of prokaryotes, mRNA is already active and these processes doesn't occur. RNA polymerase 3 transcribes tRNA, 5srRNA, and snRNA. Splicing. hnRNA is subjected to a process called splicing where the introns are removed and the exons are joined together in a defined order. hnRNA also undergoes additional process called capping and tailing. Capping. In capping, an unusual nucleotide methyl guanosine triphosphate is added to the 5' end of hnRNA.
Tailing. In tailing, adenylate residues are added at the 3' end in a template-independent manner. This is known as polyadenylation. It is now this fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation.